Hi, my name is Sudha Mesra. I am a PGY2 internal medicine resident at St. Joseph Hospital, Chicago, Illinois, and this is my submission. I'll be talking about identification of four upregulated genes in tumor tissues with adenocarcinoma of lung associated with poor prognosis via bioinformatic analysis. So, as we all know, lung cancer is one of the leading causes of death in the United States. It has the second highest incidence. The aim of this study was to identify genes which have worse prognosis for lung adenocarcinoma. Methods. So for this analysis, we use data processing of differentially expressed genes or DEGs. The data for this analysis was obtained from GeoDatabase, which is an NIH-maintained database which stores all the unique genetic identifiers for different kinds of conditions. The two databases that we accessed for the analysis was GSE 6044 and GSE 4358. So the genes between lung adenocarcinoma and normal tissue specimen were identified by an online tool to filter genes with log FC greater than 1 and an adjusted p-value of less than 0.05. So when we did this, we obtained data from 96 lung adenocarcinoma tissues and 35 normal tissues. A total of 387 upregulated genes were identified during this process. As you can see from the Venn diagram, we obtained 114 genes from 6044 and 273 from the database called 43458. Using an online Venn diagram tool, we were able to identify 24 commonly and unique differentially expressed genes amongst these two databases. As you can see in this picture, the 24 genes are listed here: SPINK1, COLA3A1, TYMS, PRC1, CCNB2, to name a few. Now, these 24 commonly expressed DEGs, we ran them through another software called David, which does a bioinformatic analysis and splits them into four components. I apologize. You will be able to see this on my abstract if you zoom in at 100%. So we divided them into biological processes, that's box 1, cellular components, that's box 2, and molecular function, that's box 3. There was also a KEG, which is Kyoto Encyclopedia of Genes and Genomes Analysis present. What we did essentially is identify the functions of these genes, like what are their molecular functions, and if you zoom in, you'd be able to see labels on each of these functions, which are arranged in the log range. So you can see some collagen fibril activation system, some peptidases, serine and protease here, some of the adhesion molecules listed. This is just the classification. We also analyze them via a protein-protein interaction network. What this does is identifies common genes and nodes and arranges them in the form of their interaction in a cellular molecule. You can see these 1, 2, 3, 4 genes which are basically not interacting with the rest of the genes. Further analysis helped us identify 4 commonly expressed genes. The top 5 and top 4 analysis of genes revealed that CCNB2, TYMS, PRC1, and CAPNA2 are the 4 genes which are overexpressed. We also did a survival analysis and a box plot analysis of these 4 commonly expressed genes. The graphs, as you can see above, demonstrate with a p-value less than 0.05 and a hazard ratio greater than 95% that these 4 genes were associated with poor prognosis in patients with lung adenocarcinoma. The conclusion of the study is these 4 genes that we were able to identify via bioinformatic analysis, namely CCNB2, TYMS, PRC1, CAPNA2, could be potential therapeutic targets for lung adenocarcinoma. They could also act as potential diagnostic markers. These are my references. Thank you so much for taking time to listen to me.